Hello, my name is Ayoob Redi and my presentation is on the topic, does pH level affect enzyme activity? Firstly, we must define what enzymes are. So basically, enzymes are proteins that speed up chemical reactions in our bodies by reducing the activation energy it takes to complete a reaction. Enzymes are important to living things because they aid in digestion, breathing, reproduction, and many other activities in our body. The enzyme that I will be using in my experiment is trypsin. Trypsin is a proteolytic enzyme made in the pancreas that is then converted into active trypsin in the lumen of the small intestine. Its function is to digest proteins. And for substrate, I'm going to use azocasein, which is a chemically modified protein and will be used in my experiment as the substrate for the enzyme trypsin. Finally, before discussing my hypothesis, I need to talk about pH level. pH level is a measure of how acidic or basic a solution is. It's ranged from pH 1, which is most acidic, to pH 14, which is most basic. So coming to my hypothesis, trypsin functions best at an optimum pH, a specific pH. Anything too acidic or too basic may negatively affect the enzyme activity. So my prediction is pH level under 7 and over 9 will negatively affect the enzymatic activity of trypsin. The reasoning behind this is that trypsin is found in the pancreas and the pH of the pancreas ranged from 8 to 8.5. So variables. My experimental variables are pH value while my control variables are salt concentration and temperature. So for my experimental design, we took 500 microliters of azocasein and 50 microliters of trypsin and added it into 5 clean centrifuge tubes. Reaction buffers were added accordingly and 500 microliters of 10% TCA was added to each tube and mixed. The tube was centrifuged for 2 minutes and then the supernatant was transferred into a clean cuvette. The spectrophotometer was set to blank at 440 nanometers using a cuvette with DH20. The absorbance was recorded and steps 4 to 8 were repeated again for the remaining 4 tubes. The experiment was finally repeated for 5 more times, but we replaced the 50 microliters of trypsin with 50 microliters of deiodinized water to get the negative control, which means seeing what the azocasein would do without the trypsin. When it comes to the results I received, they were all null results because of a fatal error in my experimental design. Discussion. My experimental design had a fatal error, which was that I forgot to incubate the sample that I was testing, and this led to all the results that came out being null. Something I would do when repeating the experiment is I would test multiple times instead of once for multiple pHs, and also refrain from adding the enzyme right away because it would work on the substrate. Conclusion. Due to a mistake in my experimental design, my data is inconclusive and no conclusion can be derived from this experiment. So basically what I mean is I don't know if my hypothesis is true or false. My references are Suji and Deniston from Department of Biological Sciences at Towson University. Thanks for listening.